What a wonderful afternoon going into the evening. This wonderful setting. Behind me is the Western Ghats. You are in the city of Delhi, which is the heart of things happening in the country. I thank the person who introduced me just now for correcting the title. She added very correctly Pentameric, Oligomeric, Bicyclo, 331 Nonanes for treating Alzheimer's and Parkinson's. Well, you could add a word there in the title disease or diseases. I repeat, the title is there are two kinds of targets from possibly the same source. The source is available in tonnage quantities again. Very important. How to choose the problem to do research work on. This source was chosen by me in March 2019 or around that time, 5.5 years ago, when I had just shifted from Regional Center for Biotechnology, Faridabad, the DBT, Department of Biotechnology, Government of India Laboratory, to the TERI-Deakin Nano Biotechnology Centre in a beautiful setting called the TERI-Gram, where there are two beautiful cricket grounds and golf courses and a wonderful stay place for a stay like a residence and a very beautiful setting. You can go around for a walk if you are allowed to enter the campus. The checking is very careful and they have some ornamental plants and so on being sold there. So one is careful. But I chose this because I understood here is a source which may not be allowed to be used for the purposes for which it is used. Coal of different varieties, koila, coal, which is also in another parlance referred to as brown coal. It's almost brownish black in color. But all over the world, including in India, the use of this particular raw material available in tonnage quantities in different places in India is being banned because it causes environmental pollution. So you'll see that when you hear more of it. But we isolated two such materials. One is, in my opinion, pentameric, the other, and oligomeric. How do we prove that? And its potential use for treating the diseases mentioned, neurodegenerative diseases like Alzheimer's, Parkinson's, or for that matter, dementia, for which there is no cure, strictly speaking, available. So this is the title. We have applied for a patent there already. We have also published one part of it, and we are doing a lot of other things on these materials. You can go to the next slide without my asking you to do so throughout the lecture. It will save some. Yes. See, why coal-based power plants are being shut down all over the world? Possibly not in the United States, but they don't want it. They make the cheapest electricity there using this process or these processes of these materials. What are the alternatives for this? Is it atomic energy, atomic power? Is it hydrogen? Is it solar energy? I do not know. And what else in the future? This concerns a material available in India in different places in large amounts. You go on to the next slide. And you see, we have claimed there is a part of the title which I should not explain now. Maybe towards the end of the lecture or in the question answer session, I will try to explain what is meant by Bicyclo Nonanes. What is meant by Bicyclo Nonanes? Okay? Don't ask me what are these materials right now. So we will talk about them at the end of the lecture. Maybe there may be a small mention during the lecture. Can these be important as some signatures of life for search of exolife? Can we look for life outside the earth of ours? And in this slide, just notice that by the year 2038, Germany has agreed to phase out coal for making power. This is all very much part of the news. People are very concerned that we don't want any pollution. The younger you are, India has met young people. The younger you are more concerned. Older people possibly have got used to it. I'm not saying they are used to it, but they possibly have seen it all their life. The younger people do not want this. So let me go back. What is this material here? Which is available in India in tonnage quantities. एक तरह का कोयला है. It's a form of coal. I told you it's sometimes referred to as brown coal. Sometimes it is referred to as leonardite. It is available all over the world. In America, in Germany, South Africa, different places. Please help me by switching to the next slide. Yes, it has multiple possible uses, which is what I want to highlight during this talk. So see, that is the code that I mentioned. You could use this material for sericulture, for horticulture, for agriculture, for biomedicine, for, as a drug carrier, a solar cell making of different varieties. So there are multiple uses that you could have for this material. We will show some of them, but though it has been studied before, but it has been clearly in the literature, it is clearly written that no chemical has been identified or characterized from it quickly. There is a patent, an Indian patent, where this material has been processed in some manner and used for growing vegetables in agriculture. We're not focusing on that. We moved away from that. Our claims are totally different from whatever has previously been done. Let me re-emphasize. No new chemical has been previously characterized from this source. And we are claiming we have more than one new chemical. Next slide shows you the isolated compound after an acidic process. This is called compound B at that time. And I think this is not my statement. Barfi was very popular in St. Stephen's College at one time. With Sukhya used to make the barfi. And this milk-based sweet. This looks like some sweet in the middle made out of milk, maybe like barfi. It is fluorescent under the UV lamp. Its original color is shown. That is black in color. How can one isolate such a wonderful colorless material from coal, so black or brown in color? And what is its use? I will tell you before we proceed further. We went one more step further and carried out an acid hydrolysis subsequently after studying the various spectra that we recorded. And we have made that second step compound last January, not 24 January, 23 January, in our unit in Pondicherry to the tune of 3.5 kilograms. Next slide will show you its mass spec of compound B. It appeared to be molecular ion of 692. 692. But that is possibly not a correct situation. We'll understand shortly. That is the first compound we isolated, which was pure white in color, as possibly a molecular weight of 692. We do not know. Next slide shows you the Next compound, which is our pentameric Bicyclo 331 nonane. Don't ask me what is the meaning of the word Bicyclo 331 nonane. Completely water soluble. You can see the solution. Absolutely clear. The solid compound is kept on the left-hand side. At that time, we used to call it A-AH. Compound A obtained by acid hydrolysis. I have proposed, plus B, even if we give you, do not drink. We will offer you a wellness drink, where a small amount of this compound is put into your Bicyclo bottle containing pure water, and you sip it or drink it, and all the diseases of the world disappear. Is this an elixir? Is it a panacea for all diseases, as I asked in the previous lecture? This material is not available anywhere in the world. ये दुनिया में कहीं नहीं है. ये कंपाउंड हमारे पास 3.5 किलोग्राम में बना के रखा है. इसमें बहुत सारा हमने स्पेक्ट्रल स्टडीज किए, हमने इसके ऊपर केमिकल थिंक्स किए हैं, डेरिवेटाइजेशन किया है. बहुत ही कीमती चीज है. कीमती इसलिए नहीं कि दाम ज्यादा है, इसके मल्टीपल यूजेस होंगे फ्यूचर में, पर हम इसको सस्ते दाम में बना के बेच पाते. We will be in a position to sell this or distribute this almost at a no profit, no loss basis. That means almost at a very low cost. Next slide. You see, it is again fluorescent under UV. It does give, as you see on the right-hand side, a kind of a purple color. But this is not as purple as I showed you the other day in the case of wall pahadians. So this hints towards the possibility that this also is an amino acid, but a sterically hindered amino acid. Next slide. This will show you, I owe this result to Professor T.N. Guru Rao of the Indian Institute of Science, Bangalore, solid state structural chemistry unit. He is now retired, but the result that is shown here is a result from the X-ray diffraction studies carried out in Professor T.N. Guru Rao's laboratory. You see the molecule, the monomer, if I may say so, the single unit. It has an amino group, it has a carboxylic acid group. Hello? Can you continue to hear me?
Yes, sir, you're audible.
Very good, very good, very good. Now it's better. So it's an alpha L amino acid. That is what I'm proposing. You know, when they isolated in New Delhi, in the Pusa Institute, Dr. S.P. Rai Chaudhary, in 1960, he isolated a compound from neem, margosa. And at that time, I don't know whether it's called AcSIR. Humorously, I call it neem, the plant neem, which is so useful to combat many diseases. Many people chew this bitter leaf early morning. They use it for, instead of Colgate toothpaste and so on. Contains a compound called AcSIR. It's not topic of our subject of our discussion today. मैं उसको कहता हूँ आजादे दरख्त इंडिया का, बिकॉज नीम के पेड़ पे कोई भी बीमारी नहीं लगती, कोई भी कीड़ा उसके पास नहीं जाता, शायद सांप भी उसके नीचे नहीं सोता, पता नहीं मेरे को. यही है वो 331 बाइसाइक्लो नोनेन. 331 अभी समझाऊंगा कि जो बाएं तरफ है, जो बीच में लाइन है ना, उसको वो तो कहते हैं ब्रिज हेड, ब्रिज कंपाउंड. वो ब्रिज के लेफ्ट में 3 कार्बन है, ब्रिज के राइट में 3 कार्बन है, बीच में 1 कार्बन. तो इसको कहते हैं 331 नॉनेन. क्यों? तो आप देखिए 8 कार्बन रिंग में है और 1 कार्बन बीच में है, बाहर वालों को नहीं जोड़ा जाता. There is a carboxylic acid group. There is an amino group. There are 2 methyl groups. There is an exomethylene group. याद रखिए प्रोफेसर TN गुरु राव एक्सरे क्रिस्टलोग्राफर हैं. सॉलिड स्टेट केमिस्ट्री स्ट्रक्चरल केमिस्ट्री यूनिट ऑफ IISc में रिटायर हुए हैं अभी, पर वो केमिस्ट नहीं है. पर उन्होंने हमें पर इसका रिजल्ट पूरा पक्का नहीं आया, बट इट वाज रीजनेबल. वी हैव इंक्लूडेड इट इन आवर इंटरनेशनल जर्नल ऑफ पॉलीमर साइंस. बड़ा इंपॉर्टेंट है, ये हमारा मोनोमर है. एथिलीन is the monomer of polyethylene. Styrene is the monomer of polystyrene. To give Bicyclo 331 nonane, if you start numbering from the carbon where there is an amino group, you may take it as position 1, then go down clockwise. There is a position 3 where there is a methyl, 4, 5, 6, 7. So 1 amino, 3, 7 dimethyl, 5 methyleno, Bicyclo 331 nonane, 1 carboxylic acid. शायद ये नाम ठीक होगा, पर हम अपने साइंस में अपने आप को ही क्वेश्चन करते रहते हैं. वी ऑलवेज कीप क्वेश्चनिंग आवरसेल्व्स. क्या सच है? क्या प्रूफ? इसमें गलती है कोई? जब हम छापते हैं उसके बाद भी सवाल पूछते हैं. This is very interesting. You should question. In Indian tradition, in our culture, we question everything, but with great respect for the person who is doing the work. We don't throw stones at him or her. We ask him questions. आप कैसे कहते हैं सच? सच बताइए, क्या है दुनिया में सच? क्या ये शरीर है? Am I merely a body which will last for a certain number of years, or will my thoughts last far beyond me? किसी एक आदमी ने कहा इंडिया में कोई 100-125 साल पहले कि though my body will go away, my thoughts will survive maybe for another 1500 years. What did Einstein say when Mahatma Gandhi was shot dead? He said, scarcely will people believe that such a man lived in flesh and blood. कोई मानेगा भी नहीं 2000 साल बाद, 2000 years hence, scarcely will people believe that a man like this lived in flesh and blood. सच्चाई ये है, आइडियाज जो आती हैं एज अ डायरेक्टिंग जो प्रोफेसर राय चौधरी ने पूसा इंस्टीट्यूट में 1960 में डिस्कवर किया था. दुनिया भर में लोगों ने काम किया, कोलंबिया यूनिवर्सिटी, ऑक्सफोर्ड, ब्रिटेन, कहाँ नहीं काम हुआ. उसके ऊपर 45 PhD 22 साल में छपी इंग्लैंड में. कितने? 45 PhD थीसिस. ये जो कंपाउंड है इसको इतना साधारण मत समझो. इसको समझना, टू अंडरस्टैंड, टू फॉलो दिस, यू नीड टू अंडरस्टैंड अ लॉट ऑफ केमिस्ट्री. हाँ, इंडिया में लोग हैं. देयर इज अ साइंटिस्ट इन बेल्लोर, PIT, Vellore Institute of Technology, who has published a large paper, very large paper, on Bicyclo 331 nonanes, how to synthesize them in the laboratory. That means these can be synthesized in the laboratory, so you don't need the natural source. पर देखिए, बाएं तरफ, if you count, 3 कार्बन बाएं हैं, ऊपर 1 है, एक नीचे 5 और बीच में 1 6. ओहो, लेफ्ट वाला 6 मेंबर रिंग है, तो राइट वाला क्या है भैया? 1 2 3 सीधे लाइन में और 3 दाएं तरफ. ओ 6. ओहो, it appears 2 6-membered rings are fused to each other. 2 6-membered rings are fused to each other, but the fusion is not 1 2, it is 1 3 position में फ्यूज हुए. अच्छा, तो ये 2 साइक्लोहेक्सेन हैं. हाँ, पर हमने तो सुना था फर्स्ट ईयर में आपसे, सेकेंड ईयर में सुना, थर्ड ईयर में सुना, MSc में सुना, PhD में पढ़ा, ये साइक्लोहेक्सेन्स तो हमेशा फ्लक्शनल रहते हैं. एक्सियल ग्रुप इक्वेटोरियल बन जाता है, इक्वेटोरियल एक्सियल बन जाता है, रिंग इन्वर्शन होता ही रहता है. अब 2 रिंग्स हों और साइक्लोहेक्सेन तो फ्लैट नहीं होता है. ये चेयर कॉन्फर्मेशन में होता है, बोट कॉन्फर्मेशन होता है, ट्विस्ट बोट कॉन्फर्मेशन में होता है। तो ये जो मॉलिक्यूल आपके सामने है, ये चेयर कॉन्फर्मेशन में रह सकता है, चेयर बोट कॉन्फर्मेशन, इट कैन स्टे, इट कैन बी बोट कॉन्फर्मेशन, इट कैन बी बोट चेयर कॉन्फर्मेशन, अगेन द रिवर्स चेयर कॉन्फर्मेशन. This conformational isomerism, or the ring inversions, will make this compound a very difficult compound to record an NMR spectrum, because NMR cannot record changes which take place at a very high speed. Next slide. Thank you for your patience on this slide. What we have proposed is this molecule as either a molecular weight of 1147 or another 32 units added. It is possibly 11, maybe around say 1189 or 1179. That's to be checked. But molecular formula यहाँ जो दिया है C69H115 N5O11. देखिए, 5 such units have led to the formation of this pentamer. And this is a new non-proteinogenic amino acid. कहाँ से मिला आपको सर? ये मिला हमें वो कोयला जो इंडिया में टन्स में है. इंडिया ने शायद कहा है कि दुनिया को बताया है कि we will stop using it well before 2050, 2045 या 2048 हम उसको कोयले को नहीं इस्तेमाल करेंगे पावर जनरेशन के लिए, पोल्यूशन नहीं होने देंगे. कितने लोगों की नौकरी जाएगी इसके लिए? हम कह रहे हैं उसी स्टार्टिंग मटेरियल से हमने एक पेंटामर बनाया है, ए न्यू नॉन प्रोटीनोजेनिक अमीनो एसिड. अगले चलिए. नेक्स्ट स्लाइड shows you its mass spectrum. We have seen this gives different results in different instruments. All over India, we have got the mass spectrum recorded. It appears here it is 1130.3. But there may be the presence of another 32 mass units, that means the CH3OH added, and another CH3OH added, और 1130 में एक OH 17 1147 हमने इसका मॉलिक्यूलर वेट पहले माना था. अब हमें पता नहीं 1147 है, 1179 है, 1189, पर ऐसा लगता है कि पेंटामर है. अच्छा, तो कब आपको पता चलेगा? जब हमें और भी ज्यादा सोफिस्टिकेटेड मास स्पेक्ट्रल इंस्ट्रूमेंट्स मिलेंगे, जहां फ्रेगमेंटेशन बिल्कुल ना हो और जहां हमें मॉलिक्यूलर वेट प्रिसाइसली हम ढूंढ पाए without any damage to the molecule. Next slide shows you. Now we are looking at the characterization data. The next slide you see its UV spectrum by left hand side. That means it is no chromophore here. Colorless to high, chromophore कहाँ से होगा? It's a flat spectrum. IR spectrum is showing some values at 1712, 1746, 1692, 1712 and 1746 are very interesting values showing the presence of some carbonyl groups. So the value of 1746 is a little higher than we anticipate for a carboxylic acid, but it is there. These are matter of facts. एक साइंटिस्ट को आपने इंसल्ट करना है तो जस्ट question his observation. If his interpretations are wrong, there is nothing wrong. But if his data is wrong, it's a very serious matter. 1712 को हम 1715 नहीं बोल सकते. हाँ, ये नहीं पता हमें क्यों इस वैल्यू पे आ रहा है. Usually carboxylic acids of aliphatic or alicyclic variety should come at about 1704, 1706. ये 1746 पे क्यों आया, 1692 पे क्यों आया, ये हम नहीं जानते. पर आजकल स्ट्रक्चर लेके यू कैन विद कंप्यूटर सॉफ्टवेयर सिमुलेट इंफ्रारेड स्पेक्ट्रा व्हिच वी हैव नॉट डन. द नेक्स्ट स्लाइड. जो बच्चों ने, जो हमारे स्टूडेंट्स ने ये टेक्निक्स नहीं सीखे हैं, वो हमें से नाराज ना हो. दे शुड नॉट लूज पेशेंस. यू विल वेरी सून बी learning these techniques and you will also be using such instruments yourself. India में बहुत बड़ी कमी है, instruments India में नहीं बनते, ज्यादातर imported. खराब हो जाए तो हमें ठीक करना नहीं आता. ये जीटा पोटेंशियल मेजरमेंट है इसी कंपाउंड का. देखो ये बता रहा है, मैं नहीं कह रहा, आई एम नॉट सेइंग इट. द इंस्ट्रूमेंट हैज गिवन अ रिजल्ट दैट द साइज ऑफ द पार्टिकल इज 6.4 नैनोमीटर्स. अरे इतना छोटा, एंड इट इज यूनिफॉर्म. दैट मीन्स इट्स अ नैनो मटेरियल. कहाँ से बनाया आपने नैनो मटेरियल? मैंने नहीं बनाया. दिस वाज मेड इन नेचर. आई ओनली प्रोसेस्ड इट. I did not make this nanomaterial. And this is a new non-proteinogenic amino acid. Next slide. ये है सर्कुलर डाइक्रोइज्म, यूजुअली कॉन्फर्मेशन देखने के लिए प्रोटीन्स के. This our compound is not a protein. It doesn't have peptide bonds, but it has 5 amino acid units. So it may have similar properties. We got a Manur Arya in the TERI School of Advanced Studies did this CD spectrum, circular dichroism spectrum. It looks like a compound which is somewhat like the peptide. It is chiral, it is optically active, and that means it may have some amount of alpha helical structure, alpha helix like DNA and peptide. Oh. But that's only an indication. It is not a confirmation. Next slide. Throughout this lecture, you are hearing me say this again and again, कि हमें सब चीजें नहीं पता. मैं कोई पहली जमात का बच्चा नहीं हूँ. I'm not a student of a first class in a school or something where I claim I know everything. I do not know. But I know if it is a Bicyclo 331 nonane, it can occupy different conformation. That means it can occupy a chair conformation. It may have a chair-boat conformation. It may have boat conformation. And all these conformations. Can you see the flat molecule on the top left? And you see the chair conformation, chair-boat conformation on the right top. And then the chair, the double chair conformation on the bottom left. And the double boat. ये सारे shapes change होते रहते हैं, पर इसमें substituents नहीं है अभी. Substituents will decide what is the energy difference between these conformations, because they will cause both steric hindrance and they will also cause what is called as electronic hindrance. इन दोनों के बीच में जो tussle है, that will decide the rate of this interchange or ring flipping. Next slide. This was again recorded by my student recently, and this has been done in another laboratory. This is the Raman spectrum of our new compound, which can be called the Pentameric compound. You see, the spectrum is very good. I hope many people will agree worldwide. There is a very interesting peak at 929 centimeters inverse. There is a peak at 1417 centimeters inverse. There is a very large peak at 2933 centimeters inverse. Yes, generally, the IR spectrum and the Raman spectrum. Raman is no other man but our own C.V. Raman, the Nobel laureate from India. And these peaks are very significant. Usually what you see in Raman, you will not see in the IR spectrum. So they are, a kind of a situation where they are complementary. And Raman shows peaks where there is symmetrical stretching or symmetrical change in the structural molecular vibrations. In this case, there could be added phenomenon of scattering. Next slide. अरे क्या बात करते हैं, अभी तक तो धरती पे थे. We were on Earth. Now suddenly we have landed on the, I hope I have not seen the surface myself. I don't know whether it's Chandrayaan or is it Mars or is it Jupiter. मेरे को नहीं पता. Some of you were not even born when I started teaching in St. Stephen's College. You were then possibly living in one of these planets. Was it Moon? Was it Jupiter? Was it Mars? I do not know. NASA ने ऐसे rovers भेजे. अब तो इसलिए नहीं भेजे. इसमें छोटे छोटे miniaturized instruments होते हैं. One of the instruments is the Raman spectrometer. One of the instruments is a mass spectrometer, very small. And the rover digs out a little of that soil, collects it so that it can return to the Earth maybe in 2030 or 2040, I don't know when. But the Raman spectral data and the mass spectral data can be collected right there and then. अच्छा, तो आपका काम के साथ इसका क्या संबंध है? यदि हमारे जो रामन डाटा है, if it is loaded on the data bank of the rover will say, oh, we have found a new compound which we have never identified previously on Earth, because we thought only 21 amino acids are involved in formation of life on Earth. We did not understand that there could be present on Earth other non-proteinogenic amino acids which might lead to the formation of life elsewhere. And there is this band which we have seen in Dr. Eswaran's work. हमने देखा है वो 992 पे, वो 1417 पे, वो वाला पीक हमने देख लिया यहाँ. ओ अच्छा, यही सेम कंपाउंड यहाँ भी खड़ा है. अरे यार, इसको कहते हैं एक्सो लाइफ को ढूंढना, आउटसाइड आवर अर्थ. हो सकता है कहीं और सिविलाइजेशन हो. अगला स्लाइड. सर, आप साइंस फिक्शन की लेक्चर दे रहे हैं कि साइंस का लेक्चर? कहानी सुना रहे हैं, ये घटना है. मैं कह रहा हूं, हमने कुछ रामन स्पेक्ट्रा रिकॉर्ड किए हैं. वी रिकॉर्डेड सम रामन स्पेक्ट्रा and we have seen some peaks. Some of these peaks are very, very significant. They are strong, and we can possibly target these for finding unnatural amino acids elsewhere in space, for that matter. So ये ऐसे चीजें, ऐसा पेपर्स अभी बड़े जर्नल्स में छप रहे हैं. There are many unnatural amino acids for biological spectroscopy and microscope. Very recent paper. मैं नहीं कह रहा हूं, हमें ये कहना पड़ रहा है because we have a new non-proteinogenic amino acid. Not one, we have so many. You are going to hear a second one today. You heard previously Gol Pahadi acid. Next slide. ताकि मैं पूरी कहानी सिर्फ एंड में ना सुनाऊं. ये वाला जो पेपर है, ये लिखा है क्या मेरे मनोरभाव, क्या आप इसके बारे में थोड़ा सा बोलेंगे? मॉडिफाइड ग्लूकोज एज ए सेंसर टू ट्रैक द मेटाबॉलिज्म ब्लो. मनोरभाव, ऑब्जर्वेशन ऑफ द 16. हाँ बताइए, सिग्नेचर पीक क्या होता है?
Yes sir. So it is related to what just previously sir mentioned about the Raman peaks. So we showed you a beautiful Raman spectra of our compound, and it shows beautiful peaks at 929 and also in the higher wave number region. So looking at this paper, they have mentioned in the
paper published, Manor Bhav, ये कब का publication है?
It is in 2023.
चलिए.
So this peak of 1602 centimeter inverse, so it is associated with the glucose metabolism. So what they have done in this paper, they have developed a sensor which could detect this, and they can identify that as a signature of life, this peak 1602. Okay, so this paper is shown here that our compound, which is showing a beautiful Raman spectra, can be used in similar way to detect life elsewhere or maybe in any system. So that's the point.
Thank you, Manarbha. Very kind of you. That means we are in a dreamy state. कि शायद हमारे स्पेक्ट्रम के जैसा एक स्पेक्ट्रम या तो मार्स या जुपिटर या कहीं वीनस कहीं नहीं कहीं देखा जाएगा. नेक्स्ट स्लाइड. Learn to dream, gentlemen, but beware of publishing your dreams unless you have tested them in your laboratory. Learn to dream, gentlemen, I should say gentlewomen, both men and women, but beware of publishing your dreams till you have tested them in the laboratory. This was a statement made by none other than Augustus von Kekule. Some students call him Kekule, who discovered the structure of benzene. He is more or less the man who started this subject of Organic Chemistry. इसमें क्या लिखा है पढ़िए जी, दोनों टाइटल पढ़िए मनोहर भाव, दोनों टाइटल पढ़िए.
Yes, so the paper on the left, its title, The Role of Chirality in the Origin of Life. So it is a very special paper written by Dr. Abdus Salam, and he was Nobel laureate in physics, and he was involved in unification of electroweak theory. That's slightly different, but what he did was he proposed a quantum mechanical explanation for why we have the levorotatory amino acids in the biological systems. So, and it has also relation to chirality. So it is a very interesting paper, and I will urge the audience, students present here, to look at it at least once time. It can be
On the right hand
slightly-
side, on the
Yes,
right hand side,
on the right hand side, this is genetically incorporated unnatural amino acids. So this is a very hot topic, we can say, that unnatural amino acids and how they can get in, how they can get incorporated into the code, natural code which is present. There are codons which are available for coding 20 amino acids, but can we engineer new codons to make new kind of amino acids? So this
which
is
journal
related to
is it published? Name the journal.
no, exactly, it was not in a journal. It was in the Biochemist Exploring Biochemist, something like that was the.
So let me explore it further. Thanks मनोहर भाव. क्या कह रहे हैं मनोहर भाव कि हमारे कंपाउंड को इंसुलिन के अंदर डाल दीजिए. इंसुलिन में 51 अमीनो एसिड है, एक जगह बीच में घुसेड़ दे दी, चाहे तो एक अमीनो एसिड को निकाल दो, और उसके बाद देखिए वो इंसुलिन डायबिटीज के लिए काम करता है कि हार्ट के लिए काम करता है, या बेहतर करता है बुरा करता है, टॉक्सिक है नहीं है. नेक्स्ट स्लाइड. देखो सबसे इंपॉर्टेंट चीज है वो सेंट स्टीफेंस कॉलेज में भी हमारे शशि थरूर जी पूछते रहते हैं, बोलते रहते हैं, बड़ी अच्छी अंग्रेजी बोलते हैं. एंड ही सेज इन सेंट स्टीफेंस वी नॉट ओनली रेस क्वेश्चंस, वी आल्सो क्वेश्चन द आंसर्स एंड देन वी क्वेश्चन द क्वेश्चंस. पता नहीं क्या क्या बोलते हैं. मतलब ये है कि सवाल पूछते रहो. कोई बुजुर्ग आदमी सफेद बाल वाला मेरा जैसे कुछ कहानियां सुनाए, उसको मत बिलीव करो, सवाल पूछो. झूठ बोलता है आदमी, शायद इसका सच नहीं है, अपना एडवर्टाइजमेंट कर रहा है. कहाँ मिलेगा ऐसा फॉर्म ऑफ लाइफ ऑन मार्स जुपिटर? हम केमिस्ट्री के स्टूडेंट हैं यार. फ्लोराइड हमें टेस्ट करना पड़ता है, नाइट्राइड टेस्ट करना पड़ता है, नाइट्रेट टेस्ट करना पड़ता है, ऑर्गेनिक है, एरोमेटिक है, एलिफेटिक टेस्ट करना पड़ता है, तब तो हमें नंबर मिलते हैं लेबोरेटरी में. आप पता नहीं क्या क्या बातें सुना रहे हैं जो हमने कभी जिंदगी में करनी नहीं है. देखो ये जो एनएमआर स्पेक्ट्रम दिखाया गया है यहाँ, दाएं तरफ ये दिखाता है, जो बड़े पिक्स को छोड़ दो, दाएं तरफ इट शोज इट इज एन एलिसाइक्लिक कंपाउंड, इट इज नॉट एन एरोमेटिक. बट इसका एनएमआर स्पेक्ट्रम स्टडी करना बहुत मुश्किल है. क्यों? मॉलिक्यूल हिलता ही रहता है. चेयर चेयर, चेयर बोट, बोट बोट, boat chair, and the other chair conformation. So it's very difficult. We have recorded this H1 NMR spectrum with some success. In most laboratories, they said we cannot record. In TIFR, we recorded, in ICG, we recorded in Delhi, and we know that it is an alicyclic compound. No aromatic portions are present in this compound. A surprising result. Next slide. But it was very difficult to record its 13C NMR spectrum. ये देखो, ये इसको कहते हैं पाउडर एक्सरे XRD पैटर्न स्पेक्ट्रम ऑफ आवर कंपाउंड, which was at that time called by us compound AAH. ये दिखाता है जो बड़े बड़े पीक्स हैं कि ये क्रिस्टलाइन मटेरियल है, अमॉर्फस नहीं है. और बाकी चर्चाएं करना हमारे मनोहर भाव हैं, हमारे सुजित हैं, उनके साथ आप चर्चा करिए. उनको मिलिए हमारी नई लेबोरेटरी में, दैट इज इन FITT IIT दिल्ली, जिसका इनॉग्रेशन हम करना चाहते हैं 14 तारीख को सुबह. आप सब आमंत्रित हैं. नेक्स्ट स्लाइड shows you, this slide shows you the studies on cytotoxicity of at concentration of 10 micromoles and 20 micromoles. टेबल में दिखाया गया है, जो बार डायग्राम है स्टैंडर्ड के साथ. आप जो जीरो, जहां 0 माइक्रोमोल है, एक और दोनों बार दे आर सिमिलर, थोड़ा सा कम है। पर आप देखिए टेबल में तो ये जो साइटोटॉक्सिसिटी माइनस की तरफ है, मतलब साइटोटॉक्सिक है ही नहीं। और कॉन्सेंट्रेशन, जितने कॉन्सेंट्रेशन अप टू 1000 माइक्रोमोल्स, बड़ा हाई कॉन्सेंट्रेशन। मतलब हमारे कंपाउंड को हम कल आपको दे देंगे सेंट स्टीफेंस कॉलेज में, खुशी से थोड़ा सा पानी में डाल के पी लेना, आपकी बीमारियां ठीक हो जाएंगी। नहीं नहीं, ये नहीं, अलाउड नहीं, अलाउड नहीं। ये पहले इंडियन काउंसिल ऑफ मेडिकल रिसर्च, द डायरेक्टर जनरल ऑफ हेल्थ सर्विसेज, एम्स एंड ऑल हैव टू सर्टिफाई व्हाट आई एम सेइंग इज ट्रू, एंड दे हैव आल्सो टू सर्टिफाई इस पीने का कुछ फायदा तो है। तो 350 रुपए की एक बोतल, उसमें 1 ग्राम हमने अपना कंपाउंड डाला है या आधा ग्राम डाला है, ये पीना ठीक है गलत है? डॉक्टर एसवी ईश्वरन और उनकी टीम को कोई हक नहीं बोलने का। वी हैव नो राइट टू टॉक अबाउट इट, वी कैन ओनली प्रेजेंट द डाटा, बट फर्दर वर्क इज इन प्रोग्रेस। नेक्स्ट स्लाइड shows you that we have been talking about, we gave this compound and all these tests cost a lot of money, in a couple of lakhs. We gave this to RCB, they took 4.5 lakhs from us. They gave very poor result. Then we submitted it to Center for Cellular and Molecular Biology, CSIR, Hyderabad, and they gave us this result that this compound does have a low 16% activity against COVID-19. पर ये साइटोटॉक्सिक है नहीं, ये कोई हार्म नहीं कर सकता है। लो कॉन्सेंट्रेशन, आप इस पानी को पी जाइए, हो सकता है वैक्सीन की जरूरत ही ना पड़े। पर बड़ा लो एक्टिव, ये एक्टिविटी 61% होती तो मेरे को आपसे बात करने की जरूरत नहीं होती, लोग उठा के ले जाते दुनिया में। अनफॉर्चुनेटली हमें 16% वैल्यू मिली, 61% होती। कोई भी वैक्सीन नहीं है जिसकी एफिकेसी इतनी हाई है। सब कहते हैं 70 है, वो है ये, पर मैं जानता हूँ 50, 55 और कुछ ड्रग्स जिसके साथ कंपेयर किया गया, जो बहुत ही पॉपुलर ड्रग्स थे, दे वर हैविंग नो एक्टिविटी अगेंस्ट कोविड-19।
रेमडेसिविर
है, हाँ रेमडेसिविर, खुद हमारे सुजीत जी ने भी रेमडेसिविर खाया है, फायदा कुछ नहीं। पर इतना नाम बड़ा, काम छोटे। हमारा नाम है
WHO, sir, WHO asked that this is failed drug.
हाँ पर वो बिक रहा था, लोग पता नहीं कितने रुपए में खरीद रहे थे उस टाइम पे। हम कह रहे हैं हमारे पास कंपाउंड है, किलोग्राम में है, थोड़ा लो एक्टिव है, तो कौन सा है, थोड़े दिन बाद ठीक हो जाएगा, 2 दिन ज्यादा लेगा। अगला स्लाइड। हमारे बच्चे जो बैठे हैं, यंग बच्चे, उससे भी एक बात मैंने कहनी, आई वांट टू से दिस। सर आप इतने टॉपिक्स में काम कैसे कर लेते हैं? नेक्स्ट स्लाइड। कर कैसे लेते हैं? आपको आइडिया कहाँ से आता है? आपके पास पैसा कहाँ से है? कितना पैसा खर्च कर रहे हैं? 4.5 लाख दे दिया उनको, 1.5 लाख दे दिया इनको, 7 लाख दे दिया उनको, कहाँ से आता है जी? और ये जो बायोसिंथेसिस, इन चीजों का होता कैसे है प्लांट्स में? क्या ये जो कोयला है उसमें कोई माइक्रोब या चूहा या बैक्टीरिया या कोई माइक्रो ऑर्गेनिज्म बैठ के ये काम कर रहा है? बिकॉज ये कोयला हजारों साल में बना है, ज्यादातर ऑफ प्लांट ओरिजिन है। डिग्रेडेशन ऑफ प्लांट्स एंड सो ऑन और टैनिंग्स ओवर थाउजेंड्स ऑफ ईयर्स। तो ये क्या कोई वहाँ लिविंग ऑर्गेनिज्म है? इज इट अ माइक्रोब? इज इट अ माइक्रो ऑर्गेनिज्म? और इज इट सम जैसे कि बड़ा चूहा हो, कोई हो, ऐसा कोई जानवर छोटा सा वहां रहता हो जो काम करता हो और ये कंपाउंड को बनाता है? बायोसिंथेसिस पर अब जैसे कि दुनिया भर में कर रहे हैं लोग, खासकर वेलोर इंस्टीट्यूट ऑफ टेक्नोलॉजी में एक ग्रुप ने बनाए हैं ऐसे बाइसाइक्लो 331 नॉनेज इन द लेबोरेटरी। नेक्स्ट स्लाइड। अरे देखो, थोड़ी देर बाद तो फिर कंप्यूटेशनल केमिस्ट्री का रिजल्ट तो आना ही है। और ये पेंटामेरिक BCNB enzymes, जो acetylcholine esterase enzyme, which is very important for studies on Alzheimer's, Parkinson, dementia. They're all different diseases, but of very similar origin. In one case, हिंदी में हम कहते थे, बचपन में सुनता था, बूढ़ा आदमी सठिया गया है। That means the proteins inside his brain have solidified and they are plaques. And somehow this should not have happened, but with age it happens. Here the binding of our compound to acetylcholine esterase, whose PDB from the data bank for X-ray results, that is the ID is 1EEA, is a very good result. Next slide. I'll show you similarly again. Next slide. That the Pentameric BCN that we have proposed in front of you was originally obtained as an ester. Then we hydrolyzed it to get the carboxylic acid. बड़ा important है. Ester हो, acid हो, क्या फर्क पड़ता है? How does it matter? It matters. Because you are saying this has to cross the blood-brain barrier. Blood-brain barrier में कुछ ही compounds अंदर जा सकते हैं 12 में ग्लूकोज है, ऑक्सीजन है, पर ऑर्गेनिक मॉलिक्यूल्स में सब कंपाउंड्स नहीं जा सकते. एंड दिस डिपेंड्स स्पेशली ऑन द एग्जैक्ट वैल्यू ऑफ हाइड्रोफोबिसिटी वर्सेस हाइड्रोफिलिसिटी. बड़ा डेलिकेट बैलेंस है. तो यदि आप ब्रेन के अंदर घुसा ही नहीं सकते, यू कैन नॉट सेंड इट इन, हाउ विल इट एक्टिवेट एंड हेल्प पेशेंट सफरिंग फ्रॉम सच अ डिजीज. All these last two slides that you are seeing. Sir. हाँ जी बोलिए.
सर मेरा एक क्वेश्चन है सर. सप्तऋषि बोल रहा हूँ सर.
हाँ बोलिए बोलिए.
Sir, I have one question. This pentameric BCN, that is a methyl ester derivative. You have said that these new derivatives are synthesized in order to control hydrophobicity. So, sir, what is its effect in the blood-brain barrier, which you have shown, sir? What is the major effect?
1 minute, मैं जवाब दे रहा हूँ. Acetyl derivative बनाए, tertiary butyl oxycarbonyl derivative बनाए. I made the methyl ester because compared to the carboxylic acid, this will be more hydrophobic. पर हमें ये अनुमति नहीं है. I don't have the freedom to, what should I say, do any studies on the brain, even in mice, or for that matter, certainly not in human beings. आप हमारे पास आइए, हम आपको सैंपल देंगे. आप जाइए नेशनल ब्रेन रिसर्च सेंटर मानेसर में, वहाँ हम साइंटिस्ट को बोलते हैं. आप जाइए ये जो बेंगलुरु में है, नेशनल इंस्टीट्यूट ऑफ बायोलॉजिकल साइंसेस, हमारे कई स्टूडेंट्स वहाँ जाके PhD भी किए हैं. आप कहीं कहीं दुनिया में हैं. तो in such places or rest of the world, go and test my compounds. I give you as many grams as you want. Test this. We have not tested. We made the compounds, we are characterizing them. But we predict, based on computational studies, that there is a possibility that this may नहीं कह रहा हूँ. बहुत सारे पेपर्स हैं. Bicyclo 331 Nonanes, आप टाइप करिए गूगल में, and Alzheimer's, Parkinson's. There are many PhD theses in America where people are saying there are some natural compounds present in nature with this skeleton which are showing anti-Alzheimer, anti-Parkinson, activity. Next slide. यार अभी तो एक पार्ट हुआ है, लेक्चर कहाँ दूसरा खत्म हुआ. अरे ये कौन, ये कुतुब मीनार है क्या? कौन लोग खड़े हैं इसको ऊपर राइट साइड में? ये प्लांट हमारे लिए बनाया गया. राइट साइड में खड़े हैं मिस्टर बालकृष्णन, who actually founded the whole thing. लेफ्ट में मैं शायद भी ठीक देख रहा हूँ, मैं पहचानता हूँ इस आदमी को. यही हैं डॉक्टर सुजीत कुमार ठाकुर, जो अभी बीच में बोले थे. मैं उसी दिन पौंडीचेरी यूनिवर्सिटी में 1.5 घंटे का टॉक दे के आया था, और ये पूरा रिएक्टर हमारे लिए बनाया गया, पायलट प्लांट. और 3.5 किलोग्राम 2023 जनवरी में ये कंपाउंड बना लिया. ए न्यू नॉन प्रोटीनोजेनिक अमीनो एसिड. सवाल पूछता हूँ, इसका आइसोइलेक्ट्रिक पॉइंट क्या है? मेरे को नहीं पता. इसका डाइपोल मोमेंट क्या है? मेरे को नहीं पता. इसके यूसेस क्या हैं? कुछ पता है, कुछ पता नहीं. शायद अलग अलग जगह यूज हो. ये हमारा प्लांट है, और ये स्टेनलेस स्टील में बना है और हमने रिएक्शन सल्फ्यूरिक एसिड में रख के भी किया है। एक और अटैच्ड यूनिट है जहाँ डाइल्यूट HCl में ट्रांसपेरेंट रिएक्टर है, वो भी किया है। ये जनवरी 2023 का। अरे क्या बात कर, यू आर अ मेयर कॉलेज टीचर, आपको किसने हक दिया कि आप 3.5 किलोग्राम कंपाउंड बनाओ? यार 1990 में, 1989, 89, 90 में both the labs in St. Stephen's College after 4 o'clock till 10:30 in the night were full of students who are preparing such compounds in their own manner in batch, process, and we produced 400 kilograms, nearly थोड़ा सा कम, आधे किलोग्राम से। पर वो सेंट स्टीफेंस कॉलेज था। जो भी वहां आए थे, all are professors in America. This inspiration is a very important student for a student at second year, first year. जब बातें समझ नहीं आती, कोई बड़ा आदमी आता है, बूढ़ा आता है, 1 घंटा बात करता रहता है, he talks many बड़ी चीजें, कुछ स्टूडेंट्स को डाउट होता है, ये सच है, गलत है, झूठ है, एडवर्टाइजमेंट। यार ये सच फोटो है, जिसने काम किया
Sir.
नहीं किया मेरे साथ। ठहरो, आपको मौका एंड में देंगे, अभी नहीं, किसी को नहीं दिया, एक सिर्फ सप्तऋषि वो अपना उसने दे दिया जरूर। नेक्स्ट स्लाइड। अरे ये ओलिगोमेरिक BCN, ये सफेद नहीं, सफेद होना जरूरी थोड़ी, पर काला नहीं। अच्छा, इसका जो मॉलिक्यूलर वेट क्या है? नेक्स्ट स्लाइड। इसका एक तो ये दिखा रहे हैं जेल परमिएशन क्रोमेटोग्राफी। आपके डॉक्टर अजीत सेठ हमारे जो फॉर्मर स्टूडेंट थे, कल भी थे, पहले भी थे, उन्होंने मेरे को मदद करने की कोशिश की, पर इंडिया में जहां भी इंस्ट्रूमेंट्स हैं सब खराब है। एक NCL पुणे में, उनसे मैंने पूछा, कहते हैं हम सिर्फ इसको टेट्राहाइड्रोफ्यूरान सॉल्वेंट में यूज करते हैं, तो हमका पानी में कर दीजिए। नहीं करें। तो ये किसी प्राइवेट कंपनी में उन्होंने पानी में किया, वाटर में किया। कैन यू सी, इफ यू कैन सी द पीक, it is only centered around one particular time point. There are 2 small peaks on the top. That means 2 very similar forms are present, and this shows a molecular weight as written here, which I'm sure you can read. There are MW is around 3907. This is the other molecule that is not the value that I wanted. ये before hydrolysis का है, और ये हाइड्रोलिसिस करने के बाद 2990 के आसपास, 2200 के आसपास आ जाता है। मतलब उसमें 223 का एक यूनिट, हमारा मोनोमर 223, 223 into 5 is around 1115, and then 1115 plus 32 is 1147. Another 32 maybe goes to 1179, and maybe up to 1190 or 89, etc. So we do not know exactly, but ये दिखा रहा है इंस्ट्रूमेंट कि 2 बहुत क्लोजली सिमिलर चीजें हैं, और उसका मॉलिक्यूलर वेट दे रखा है कि अंदाजा 3000 के पास। That means 13 such units, 10 to 13, 223 into 10 is 2230. If it is 13, it comes nearly 2900. That means it has species in the polymer, oligomer, with molecular weight range between 2200 by Mn and Nw possibly higher. So we may calculate around 10 to 13 units of the monomer. Next slide. अरे ये UV spectrum ऐसा, अरे it's very unusual. I am almost not wanting to believe it, because you see so many humps. हाँ, एक बात तो है, it was not white in color. So unlike the pentameric BCN, this seems to have chromophores, leads to color, which could have many potential uses. Molecules with such a UV spectrum could be used for making what is called dye-sensitized solar cells, which are used now commercially available for indoor lighting and charging your laptop or your mobile, etc. Next slide. We are closing in on the hour. It's Fourier transform infrared spectrum. Again, you can read the values. If you see there is a value around, I can read some value around 1405, 1554, 1635, and a small peak at about 1700. जो carboxylic acid की जो traditional values होती हैं, we are not able to see them. Maybe you can see it in the Raman spectrum. Maybe somewhere these are, not symmetric vibrations. These may be asymmetric vibrations. I do not know. But the value is not of a typical carboxylic acid. That means they may be hindered. Next slide. We'll keep to the hour. Again, the circular dichroism was done recently in water. What does it show? It shows that, that maybe we can simulate it one day. Then we'll know whether it is showing alpha helical character or it doesn't have any alpha helical character. Is it behaving like a peptide? No, it's not a peptide. And can we understand its secondary structure, not only the primary structure? But please understand, this is not a peptide. This is not an oligopeptide. This is not a polypeptide. This is not a protein. But it can be called a non-proteinogenic amino acid. Next slide. Particle size works out to 19 nanometers. Again, nanoparticle. Sir, kya baat hai? You join TERI-Deakin Nano Biotechnology Centre. So everything you make turns out to be a nanomaterial. मैं थोड़ी कह रहा हूं, I'm not saying this. This is a result from an instrument. And nobody questions the result from an instrument because instruments do not tell lies. हाँ, कभी कोई इलेक्ट्रिसिटी फ्लक्चुएशन हो तो गलतियां हो सकती हैं, पर ये आप रिप्रोड्यूस करिए 4 बारी, 5 बारी, 6 बारी, 10 बारी, सेम वैल्यू आनी चाहिए. Oh, again this is a nanomaterial. It's an oligomeric Bicyclo 331 nonane which is substituted with 2 methyl groups, a CH2 and NH2, a COOH, and it's an amino acid. Go ahead. But I may say, this is a Raman spectrum of this material. There are 2 very clear peaks. One again at a value of 2935, which is obviously a CH3 vibration, but of a symmetrical variety. And this 99929 peak, which is very significant, I would say this can be used as a signature of life based on such non-proteinogenic amino acid or acids yet to be discovered outside Earth or on Earth. अभी हमारा दोस्त कह रहा था मनोहर कि देखिए इसके लिए कोडॉन्स अलग होंगे, इसके लाइफ फॉर्म्स अलग होंगे, और ये हमारे ह्यूमन बीइंग के या जो लाइफ फॉर्म्स हम जानते हैं उससे रिलेटेड नहीं होगा. शायद ये प्लांट बेस्ड डीएनए होंगे, नए वैरायटीज के जो स्टडी नहीं किए गए. एक दिन मैं सेंट स्टीफेंस कॉलेज में अपने घर से निकल रहा था तो 500 लोग एक आदमी के पीछे आ रहे. मैंने देखा कौन आदमी बताइए? हरगोविंद खुराना. वो किस लिए सेंट स्टीफेंस कॉलेज आए? Because such a large number of people came to hear this noble orate. The space in the university was not enough. They walked up to the principal of St. Stephen's College to explain and ask him for permission to hold a lecture by Professor Hargobind Khurana in St. Stephen's College. Everybody came for the university. यार 24 घंटे में से 19 घंटे काम करते थे हरगोविंद खुराना. नोबेल प्राइज मिला. 19 out of 24 hours on the day he was given the Nobel Prize, they had to see where he is. He was hiding inside his laboratory underground so that nobody disturbed him. ऐसे लोग ही इनाम पाते हैं, नाम पाते हैं, काम करते हैं. सिर्फ बात करना नहीं, हमें लेबोरेटरी में काम करना पड़े, कंपाउंड्स अच्छी मात्रा में बनाना पड़े, पर अपने स्वार्थ के लिए नहीं, एक निष्काम भाव से दुनिया को सफरिंग को कम करने के लिए सेवा भाव से. और यही ट्रू स्पिरिट ऑफ द क्रिश्चियन of St. Stephen's College. We have not worked with huge budgets. St. Stephen's College has worked all along on a very shoestring budget. पैसा ही नहीं था. बिना पैसे के ऐसे उच्च कोटि के स्टूडेंट्स प्रोड्यूस किए, बहुत बड़ी बात, और आज भी कर रहे हैं। आप में से जो मेरे को सुन रहे हैं, एक एक पोटेंशियल वर्ल्ड विनर हैं। मैं ना देखूं आपको, पर दुनिया देखी। अरे भाई, ये टीचर्स कौन थे जिन्होंने इनको इतना प्रोत्साहन दिया? Who have encouraged them, who inspired them. The very building of St. Stephen's College inspires you. Although we were previously in Kashmiri Gate, this building came up in 1939. Those of you who are staying in the campus, you know it's a very old building. I understand even the roof is giving way, but it's a building of great national importance. Mahatma Gandhi used to come and stay in the house of the principal of St. Stephen's College when he was well known, but the British police and British government was trying to arrest him and put him behind bars. So the principal of St. Stephen's College was brave enough to receive Mahatma Gandhi in his home. आगे जाइए सर। आप ये महात्मा गांधी की कहानी कहाँ से ले जाते हैं? आप तो ओलिगोमेरिक और पेंटामेरिक बीसीएन की बात कर रहे हैं यार। इस एनएमआर को छोड़ दो, अगले में चलिए। किसी ने जिसने भी बनाया, बड़ा चीज बहुत अच्छी बनाई है दाएं तरफ। ये भगवान ने बनाई, ये नेचर ने बनाई, और ये 3D स्ट्रक्चर है। थोड़ा सा आप देख सकते हैं कि एस शेप है, ट्विस्टेड है, हेरिकल है, और ये पेंटामेरिक नहीं है, ये ओलिगोमेरिक है। नंबर ऑफ यूनिट्स ऑफ द मोनोमर इज 10 to 13. Who is saying this is 10 to 13? I am not saying it. It is the gel permeation chromatography which is giving us the proof. Nobody will question it. And most of the instruments, gel permeation chromatography instruments bought with the government money in most national laboratories are not functioning. Why is this happening? यार हमको दे दो, हम सेंट स्टीफेंस कॉलेज में उनको ठीक करके रख लेंगे। चलिए अगला स्लाइड। और कॉलेजेस के पास तो कुछ भी इंस्ट्रूमेंट्स नहीं। ये देखिए मॉलिक्यूलर डॉकिंग of this oligomeric BCN on different, receptor sites, particularly this, receptor type 3 PDB number is given, and so beautifully our compound is going and sitting inside the receptor binding site. इसकी बाइंडिंग एनर्जी हमारे स्टूडेंट्स बताएंगे आपको। लग रहा है बहुत अच्छी तरह से बाइंड हो रहा है। तो मतलब क्या है? ये शायद हमारे लिए बायोलॉजिकल एक्टिविटी जो मैंने आपको जिक्र किया है, which I mentioned, namely towards Alzheimer's, towards Parkinson, and maybe even as an anti-cancer compound, who knows? I am not saying this. This is docking study done with molecular, the kind of computer software that's available today, which is free. I don't have the money. Once we bought with CSIR grants, we bought Schrödinger software for 3 years. At that time, there are not many students who are very well versed in it, but Stephanians are now, and many other students in many other colleges have compulsory papers in second and third year. I do not remember it's correct or not, but certainly they have computational chemistry. So they are quite well versed. For this, you do not need any chemical, you don't need a lab, you need access to a computer, your own personal computer, and you should have dedicated software and dedicated computer, dedicated time. Computer time. Next slide. We've just crossed the hour, I know, so we should close. Similar results are with this PDB, which I read as 7BQX, which is a intestinal peptide receptor, vasoactive intestinal peptide receptor. That means यहाँ भी इसकी activity हो सकती है. Who is saying this? A molecular docking results with computational software is saying. Next slide. You remember, I went to Lindau. At that time, these 3 Nobel laureates were there. 1 or 2 of them have subsequently died. At that time, in the middle you see a green material. That is the green fluorescent protein. It's very highly talked about because you could study many biological processes. If you tag a molecule with the green fluorescent protein, one of these 3 Nobel laureates was able to produce not green, but all colors that you can imagine. One from Japan got it originally isolated. Man in the middle was able to carry this work further. All the three shared the Nobel Prize. I met them in person, I photographed with them, talked to them. Now, our oligo BCA, you can come and see it in our lab, shows green fluorescence. Try to tell you, ask my students what is the cost of a small amount of green fluorescent protein today in the market. Huge price, very difficult to get. Next slide. These are the Nobel laureates with whom my wife is on my right, and this is that particular Japanese professor, Nobel laureate Shimomura. His wife said to me and my wife, "Dimag kharab ho gaya tha." He is not able to answer coherently. The wife was telling us in the Nobel Prize winners meeting in Lindau, the other Nobel laureates are also mentioned and shown in the next slide, adjacent slide in Lindau. I hope some Stephanians will get selected. When I went second time to Lindau, two of our students from St. Stephen's College Chemistry Department were representing India. But I was not representing India alone. I had been invited second time by the German, organizers as one who had already been to Lindau conference, Nobel Prize winners conference in 1977. So once again, I was given this chance. It's a great, unique honor where you are sitting with Nobel laureates and their spouses. Many people were not sure whether I was a Nobel laureate or my wife was the Nobel laureate. And my wife felt very happy because nobody could find out who is the Nobel laureate. And quite likely she was the Nobel laureate. And therefore, we are ending this lecture on a very memorable note as we conclude. Next slide. क्या ये एंटीबैक्टीरियल प्रॉपर्टीज दिखाएंगे? तो ऐसा करें ना, इसका एक टी शर्ट बना देते हैं, सेंट स्टीफेंस का स्टार लगा के, वो यंग पीपल यदि धो ना पाए तो भी उसमें बैक्टीरिया नहीं ग्रो करेंगे. हम सोच रहे हैं, we are thinking, should we try and make in the next 2 years some t-shirts or hankies which are coated with our material, and it shows, first of all, somebody has to find out what is the antibacterial property. I'm not the person to do so. On the left-hand side, there is a paper using poly-L-lysine. You can go to the Google and see what is poly-L-lysine. You can see this paper, you can read. Our compound is very much like poly-L-lysine. Lysine is an amino acid, one of the 21 amino acids known in the human body. But here, we have something which we have isolated, and we claim there is a chance that this may show antibacterial activity. Future alone will tell whether it is so, yes or no. Next slide. July, you can see that we have synthesized, this is inside the flask. Dr. Sujit Thakur came to Pondicherry via Coimbatore, discussed with me, enjoyed the surroundings here, and went on to the Pondicherry lab, and he produced 100 grams of this compound. यदि मैं उनसे पूछूं 3 किलो बना सकते हो? हाँ जी, सारा चीज दे दीजिए, 1 हफ्ते में बना दूंगा. नेक्स्ट स्लाइड. हमारे पेटेंट जो हमने अप्लाई कर रखा है, पब्लिकेशन जो इंटरनेशनल जर्नल ऑफ पॉलीमर साइंस में है, और उसके साथ साथ वी वांट टू आल्सो सी इफ वी कैन publish another paper of the Oligo BCN, and we can do further work on these compounds. Next slide. I think that's the end of this talk, which is just a shade over an hour. Generally, I should not talk more than an hour. Maybe I've crossed 4 or 5 minutes beyond that, for which I apologize to the audience, but I'm ready for any questions or comments from anyone of you.
Sir, another question from my side.
Yeah.
Sir, can we find any kind of molecule in Parkinson's disease that will help to solve the disease. And also because NABL lab, you might have heard of many national accredited labs, they are working on Parkinson's disease. But sir, we are really very worried that this disease is not having any kind of proper treatment. We are not, computational chemists are not being able to generate, and continuously we are seeing that there is a tremendous growth in this disease. Is there any solution to stop or curb this disease?
I will answer a question in brief, not in detail. पहली बात है Alzheimer's और Parkinson's अलग बीमारी है, but है तो दोनों brain की. They are very related to each other. In Parkinson, your amount of L-DOPA, L-dihydroxyphenylalanine released, which gets converted, down the road into serotonin and neurotransmitters, etc., their amounts change and decrease. So that's a problem. The question is also that the proteins become flakes and they are no longer active. They are not able to interact with each other. They are not flexible. They become hard. And these can be studied. Now, one small advice to Saptarishi, which is also an advice to everybody else present. The computer से करके नई दवाइयां नहीं डिस्कवर नहीं होने वाली. कंप्यूटर आपको रास्ता दिखा सकता है, पर रास्ता, the road, you have to cover the road. आपने रोड साइन देखा, बोर्ड देखा कि यहाँ से आप कनॉट प्लेस सीधे जाइए, 10 किलोमीटर है St. Stephen's College. I don't know how many students would want to walk from St. Stephen's College to Connaught Place. How many would walk to Kashmiri Gate? I have done this. One of our former students, Pradeep Mehta from the 1973 batch, one day just walked up to my house. He said, sir, I had my police vehicle. He was an IPS. I have my assistants with me. I just walked from Connaught Place to St. Stephen's College. Sir, I wanted to meet you. His 1973 batch. कौन चलेगा? Lifestyle क्या है? 60 kilometers, 80 kilometers, 120 या 200 kilometers में आप कार चलाइए, 2 लोगों को मारिए और उससे सारी बीमारियां ठीक नहीं यार. कंप्यूटर नहीं सॉल्व करने वाला बीमारियों को. इट्स अ वार्निंग. रास्ता दिखा सकता.
As we reach the penultimate day of our lecture series, we thank you, Professor Eswaran, sir, for delivering an enlightening and engaging lecture today. Your insightful expertise and passionate presentation have inspired us all. We deeply appreciate the valuable time and effort you invested in sharing your knowledge with us. Thank you once again, sir.
Thank you so much. You speak so well, all of you. Wish you all good luck.